Hello classmates, my name is Mark Gilkerson and I've put together this video to help explain the general concept of absorption spectroscopy. The following will be a general overview of how this absorption spectroscopy works and how the technique is performed. Some of you may be familiar with the acronym ROY-G-BIV. ROY-G-BIV represents the visible component of the electromagnetic spectrum and stands for the colors in the order of their wavelengths from longest to shortest: red, orange, yellow, green, blue, indigo, and violet. This, however, is not the entirety of the electromagnetic spectrum, but a narrow portion of it which is visible to the human eye. Also in the electromagnetic spectrum are radio waves, microwaves, infrared, ultraviolet, X-rays, and gamma rays. Radio waves are anywhere from 10 to 1,000 meters in length, and gamma rays are 10 to the negative 10 meters in length, very small. Now, the visible portion of the spectrum, the ROY-G-BIV part that we can see with our eyes, exists in the middle of these two in the 400 to 700 nanometer range. When electromagnetic waves hit an object, some areas of the spectrum are absorbed while others will pass through. As a result, electromagnetic waves which can pass through are observed, and those that are absorbed by the object are left out of the observable data which is collected in an experiment. This diagram illustrates the general setup which would be used to conduct absorption spectroscopy analysis of a sample. In this example, a light source is used to send EM waves through a monochromator, which is an optical device which transmits a narrow, selected band of EM radiation drawn from a larger range of wavelengths. This selected band of the spectrum is passed through a sample, such as a gas or suspension of proteins in a solution. As this wave passes through the sample, some parts of the spectrum are absorbed by the sample, while others will pass through to the detector on the other side. The detector feeds the information it gathers about the portion of the wavelengths it picks up to a device, which it will amplify and display the information. The information displayed graphically can show the spectrum recorded by the detector, as well as illustrate which parts of the spectrum were absorbed by the sample by showing those areas as black bands, as we can see here. Now, in this depiction, these individual black bands represent parts of the EM spectrum which were absorbed by the sample here and never made it through to the detector for us to observe on the display over here. These areas directly correspond to the parts of the wavelength which are absorbed, and based on the absorption that occurred, the composition and structure of the sample can be determined based on the patterns created by known substances. The bottom illustration depicts a sample containing one peak of absorption around 480 nanometers on the graph plotting wavelength versus absorption. Though some substances will have many areas of absorbance, such as in the top illustration here, where there are 8 separate distinct bands representing individual peaks on a graph like the one below. If this information here were to be represented on this graph here, you would see 8 peaks instead of just the one peak here. And if this were represented on this here, there would just be a single band of absorbance. This has been a general review of the basic concept of absorption spectroscopy, and more information will be provided in the rest of the presentation. Thank you for listening.